Chapter 10, the structure and function of DNA. Why molecular biology matters. DNA is the molecule of heredity and the chemical basis for evolution. Its structure and function are therefore among the key topics in the study of life. While learning about DNA, you'll explore some of the most important processes that underlie every inborn trait. In the year 2015 in Brazil, it was found that a large number of babies were born with damage to their nervous system and sense organs. These babies had neurological problems, they had difficulty in eating, there were muscle and joint problems. So it was later revealed that these abnormalities were related to exposure to a new virus called the Zika virus. And then in the year 2016, the WHO, World Health Organization, had to issue a worldwide health emergency for Zika. Now, literature analysis showed that Zika was not a new virus. It was first discovered to be infective to human beings in 1952, and usually it is not dangerous to healthy adults. Because it is not very well researched, there are very few weapons or treatments against Zika. There is no vaccine, and we only use medicines to manage the symptoms. Like all other viruses, the Zika virus has a very, very simple structure consisting of a nucleic acid molecule. Therefore, in order for us to fight against any virus, we need to understand its life, its mechanism of reproduction, how it divides or it increases in number at the molecular level. This is how the Zika virus looks. This brings us to the genetic material, DNA, and how, what is its structure and how it replicates. By late 1800s, it was already known that DNA was a chemical component of cells. However, Mendel and other early geneticists had no idea of what was the DNA's role in the cell. Fast forward to 1950s, and a series of discoveries occurred which convinced the scientists that DNA was the molecule that was serving as the hereditary material in the cells. Not much was known, but still enough to convince the scientists that it had the capacity to store genetic information, it could copy the genetic information and then transmit it to the next generation. So what are DNA and RNA? Both DNA and RNA are made up of nucleic acids. These are polymers which are made up of single units called monomers. Polymers means long chains, and these monomers are called nucleotide. Therefore, both DNA and RNA are polynucleotide. That means these are long chains made up of those nucleotides. So imagine a child playing with Lego blocks and has created a long tower. So that entire tower is the DNA, whereas each Lego block is a nucleotide, that is the monomer. And these nucleotides are represented as or abbreviated as A, C, T, and G. There are 4 types of nucleotides. And the nucleotides are joined together by means of covalent bonds. You've learned about this in one of the previous chapters. And this covalent bond is between the sugar of one nucleotide and a phosphate group of the next. And this pattern repeats. So there's a sugar, phosphate, and this pattern is known as the sugar phosphate backbone. Each nucleotide consists of 3 parts or 3 components: a nitrogenous base, a sugar, and a phosphate group. The nitrogenous bases are arranged like ribs which are projecting out from the backbone. In DNA, the sugar is called deoxyribose, while in RNA it is ribose. So deoxyribose is missing 1 oxygen atom. It has 1 oxygen atom less than that of ribose. The full name for DNA is deoxyribonucleic acid. You look at the D, the N, and the A, while the full name for RNA is ribonucleic acid. This figure illustrates the chemical structure of a DNA polynucleotide. The 4 types of nucleotides present in DNA, they differ in their nitrogenous bases, and they can be divided into 2 main types. The first type are thymine and cytosine. Thymine is T, cytosine is represented by C. These are single ring structures, and these are known as pyrimidines, while adenine and guanine, A and G, these are larger double ring structures and are known as purines. In RNA, there is a difference. Instead of the thymine, there is uracil or U. And RNA also contains a sugar that is different from DNA. In DNA, it is deoxyribose. In RNA, it is ribose.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
The structure of DNA was discovered or given to the world by 2 scientists, James Watson and Francis Crick. They determined that the DNA is a double helix. It is a helix with a uniform diameter, and it is made up of 2 polynucleotide strands. Behind, it is said that behind every successful man there is a woman. So behind these 2 successful men was also a female scientist. So these 2 scientists, when they were visiting the laboratory of Maurice Wilkins at King's College in London, Watson saw that there was an X-ray image of DNA which was produced by Wilkins' colleague named Rosalind Franklin. And it had taken her several hours to get that very, very precious image. So just looking at that image, Watson and Crick, they started concluding what the structure could be to produce that image. So they used wire models and they began to construct a double helix that conformed to all known data about DNA. These are the discoverers of double helix. On the left side, you see Watson and Crick, and on the right side is Rosalind Franklin. The DNA structure is like a spiral staircase. The sides or the railings represent the sugar phosphate backbones, while each rung or each step represents a pair of nitrogenous bases which are connected by hydrogen bonds. And there's a particular base pairing rule that dictates the side-by-side combinations of bases that form the rungs. A always pairs with T and C always pairs with G. Remember this as apples grow on trees and cars go into garages. So A with T, adenine with thymine, and cytosine with guanine. This is a rope ladder model of the double helix. This slide gives you the descriptions for the diagrams or the images, figures that you would be seeing in the next slide. On the left side is a ribbon-like diagram showing you the DNA structure. In the middle is a more precise chemical atomic model of the DNA showing you the hydrogen bonds. while on the right is a computer-generated model.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
Watson and Crick published their findings in 1953, and they received the Nobel Prize in Medicine for their work in 1962. Unfortunately, Rosalind Franklin died of multiple cancers in 1958 and thus could not receive the Nobel Prize, which is given only to living scientists. Watson and Crick's DNA model shows that whenever a cell is reproducing, a cell has to divide. The DNA would serve as a template. Each strand of DNA would serve as a mold and it would guide the reproduction of the other strand. And how would the other strand reproduce? By following the base pairing rule. This figure shows DNA replication. The blue colored is the parental old strand, while the new strand is shown in brown or golden. So it is just that the new daughter strand will be formed by following the base pairing rule, where A pairs with T and C pairs with G.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand, while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
Replication of a DNA molecule begins at a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in 2 daughter DNA molecules. Let's now look at the process of DNA replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The 2 strands would naturally tend to rewind, but are held apart by molecules of a single-strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction. The strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview primer, shown here in red, towards the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary DNA nucleotides to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA. Next, the enzyme DNA ligase links the Okazaki fragments to form the lagging strand.
DNA replication is carried out with the help of several enzymes, which you've already learned in the animations from the previous slides. You've learned about the replication bubbles, the origin sites, the DNA polymerases, and other enzymes. There could be multiple bubbles in a replicating DNA.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
The flow of information in a cell and in an organism as a whole goes from DNA to RNA. RNA is like a photocopy of the DNA, and then to protein. This is known as the central dogma. So DNA is very precious. It contains all of the genetic information. It provides instructions or orders to the cell and the organism as a whole. The organism's genotype, which is the genetic makeup, the genetic constitution, is all of the heritable, hereditary information contained in the sequence of the genes in the DNA. While the organism's phenotype, which are the external features, the physical traits, they arise from the type of proteins, the expression of the protein. So it is the DNA, which is the genetic makeup, it will decide what color the organism is going to, a color of eye the organism is going to have, or what color the organism's hair, will be. And then, depending on the DNA sequence, the RNA is made, and then depending on the RNA, the protein will be made, and it is the protein that will eventually give rise to that particular hair color or eye color. So let's get some more technical details about this flow of information. So first step in this flow of information is DNA replication, where DNA just creates a copy of itself. The second step is when an RNA is made from the DNA, that is known as transcription. And then the third step is when a protein is made from the RNA, that is known as a translation. This figure shows transcription and translation. Transcription occurs within the nucleus, while translation occurs in the cytoplasm. So how are proteins made from DNA? the language has to change from nucleotides to amino acids. Amino acids are the building blocks of proteins. A gene is a segment of DNA and it has thousands of nucleotides. So when DNA makes RNA by the process of transcription, it is still the language of nucleotides. It's just RNA is making a photocopy of the DNA. And then when RNA is translated into protein, that's where the exchange of language occurs from sequence of nucleotides to a sequence of amino acids. And this is facilitated by the RNA molecule. And this linear chain of amino acids is called a polypeptide, which is a simple protein. It is known that the flow of information from gene to protein is based on a universal triplet, that means a 3-letter code. So the instructions for the amino acid sequence of a polypeptide chain are written in the nucleic acids in DNA and RNA as a series of 3 base words. So that triplet code is made up of 3 base words, and each word is a codon. So these 3 base codons in the DNA will get transcribed into complementary 3 base codons in the RNA, and then the RNA codon would be translated into amino acids that will form the protein. This process shows how the blue-colored DNA strand would be transcribed into the red-colored RNA strand, and then 3 bases of that RNA, which constitute 1 codon, would make 1 amino acid. So the genetic code is a set of rules that convert a nucleotide sequence in RNA to an amino acid sequence in a polypeptide. Various permutations, combinations show that 64 triplets can be made, and out of those 64 triplets, 61 code for amino acids, while 3 are stop codons, which are like stop signals, and they instruct the protein making factories, the ribosomes, to end the polypeptide. So one RNA triplet would always specify one particular amino acid. This is the genetic code listed by the RNA codons. You have to start from the center of this circle. So the first alphabet that you see, that is the first base, so G. And then if you go to, from G, if you go to A, and you see that both GAC and GAU would code for aspartic acid. If you see the green colored one, this is methionine, this is the start codon. You also see the 3 stop codons shown by red. The genetic code is almost universal and is shared by all different types of organisms, from the simplest bacteria to the most complex human beings. And this universality, it shows, or it suggests that evolution occurred, and all organisms, they started from a single common ancestor. And because of this common genetic code, it is possible to maneuver one species to produce a protein from another species. So genetic engineering is possible because of this universal genetic code. So transcription is the process of formation of RNA from DNA by following the base pairing, the complementary base pairing rule. So transcription starts from DNA, where one strand would serve as a template, and nucleotides that would make up the new RNA would add one at a time, forming hydrogen bonds with the DNA nucleotide bases there. Now, please remember that in RNA, there is no thymine. It is instead substituted by uracil. So that means instead of a T pairing with A, it would be a U pairing with A, and the enzyme that is used to link the RNA nucleotides is known as RNA polymerase. The start signal for transcription is a nucleotide sequence called promoter, which is present on the DNA at the beginning of a gene, and this is where RNA polymerase would attach. Transcription involves 3 steps: initiation, elongation, and termination, and we will learn about this in the animation. Transcription stops in the last phase, termination phase, when the RNA polymerase enzyme, it reaches a particular termination signal called the terminator, and then the polymerase, it detaches from the RNA and the DNA strands rejoin. This figure depicts the 3 phases of transcription: initiation, elongation, and termination.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
This is the completed RNA and detaches from the DNA.
When the RNA has been transcribed, the transcription occurs in the nucleus. Now this RNA is known as moved into the cytoplasm. But before it moves into the cytoplasm in eukaryotic cells, this RNA needs to be processed. And there are different types of processing that occur. One of them is addition of extra nucleotides to the end of the RNA transcript. And these additions, called the cap and the tail, these make the mRNA ready for export from the nucleus and then help the ribosomes which are in the cytoplasm to recognize the RNA as mRNA. Another type of processing that occurs in the RNA is deletion of the non-coding sequences in eukaryotic organisms. So most genes of plants and animals, they include these internal non-coding regions which are called introns, and the introns are interspaced in between the coding regions, which are the parts of genes that are expressed in proteins, and those are called exons. So when RNA is initially transcribed, both exons and introns are transcribed from DNA, but this is a really long sequence, and before the RNA goes out of the nucleus, the introns are removed, they are deleted, and the exons are then joined to produce an mRNA molecule with a continuous coding sequence. And this cutting and pasting process is known as RNA splicing. This figure shows RNA processing. It is depicting the splicing of introns, rejoining of exons, of the cap and the tail. Now we've learned all about RNA means making. So this is conversion between different languages, from nucleic acid language to protein language, and this is much more complicated, more elaborate. It requires more elaborate machinery than transcription. So the first ingredient that is needed for making proteins is, of course, the mRNA. In addition to mRNA, there would be enzymes, there would be energy requirement, and ribosomes, which are organelles present in the cytoplasm, as well as another type of RNA called transfer RNA or tRNA language to talk to the protein that it's going to form, which needs the amino acid language. So to perform this task, the tRNA molecules must carry two distinct functions. First, they should be able to pick the appropriate amino acids, and second, they should be able to recognize the appropriate codon on the mRNA. A tRNA molecule is made up of 80 nucleotides of RNA and twisted and folded upon itself to form an inverted L shape or a clover leaf. During phases, and on the other hand, the tRNA molecule has a site which a particular type of amino acid, only one type of amino acid will attach Although all tRNA molecules are similar, there are slightly different versions of tRNA for each type of amino acid that it will carry. This figure shows the structure of tRNA with the anticodon and the amino acid attachment site. Ribosomes are organelles in the cytoplasm that serve as the protein-making factories where they coordinate the working of mRNA and tRNAs and actually make the protein. A ribosome has two subunits. Each subunit is made up of proteins and a special type of RNA called ribosomal or rRNA. A fully assembled ribosome, the binding for mRNA is a small subunit, while that for ribosome, just like transcription, translation also has three phases: initiation, elongation, and termination. Initiation involves coming off the mRNA, the first amino acid with its tRNA and the two subunits of the ribosome. The mRNA molecule has the cap and the tail that would help the mRNA bind to the ribosome. This is a molecule of mRNA. Initiation step involves two substeps. First, the mRNA molecule would bind to the small ribosomal subunit, and then a special type of initiator tRNA would bind to the start codon where translation begins, okay, on the mRNA. The large ribosomal subunit would bind to the small one, creating a functional ribosome. Once initiation is complete, then amino acids are added one by one to the first amino acid each. This figure shows the initiation of translation. This figure shows the elongation of a polypeptide. Elongation goes on until a stop codon or a stop signal is reached at the ribosome's A site. So the completed polypeptide would then be freed and the ribosome splits back into its subunits.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into three stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
This slide describes legends for figure on the subsequent slide, and it reviews the flow of genetic information in the cell, that is the central dogma from DNA to RNA. This is a summary of transcription and translation.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increased amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. The instructions for making insulin are coded by a segment. Enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA and processed in several ways. First, a modified guanine nucleotide. Also, segments of the RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.